So in 2018, excuse me, Cabaletta was formed with one purpose: to develop and launch the first curative, targeted cellular therapies for patients with autoimmune diseases. We're doing this by pursuing two strategies in parallel: the CAR-T strategy and the CAR-T strategy. The CAR-T strategy is CAR-T for autoimmunity, with a lead program, CABA 201, which recently had its IND cleared by FDA for the treatment of lupus and lupus nephritis. CABA 201 is quite an exciting product because it's been specifically designed for treatment of autoimmune patients. It has the same 4-1BB co-stimulatory domain and a similar binding affinity as the binder that was used in the recent publications in the New England Journal of Medicine, Nature Biotech, and the Lancet Rheumatology. All three of those publications showed clinical complete responses in total in a half a dozen patients with lupus and with myositis. Quite remarkable findings. CABA 201 has a fully human binder, and that fully human binder has been studied in approximately 20 patients in oncology setting, and the binder has excellent safety and tolerability, which I'll discuss a bit more later on. The characteristics of CABA 201 supported the clearance of an IND with a very efficient clinical trial design. And specifically, what I'm referring to is our belief that rather than starting with a lowest dose and slowly titrating up over a period of many months, perhaps years, we believe that we may be starting with a fully therapeutic dose. In addition, the dosing interval between patients has been shortened appropriately based on the characteristics of the product and the filing that we've made. All of these characteristics are going to be applied into our lupus study, which studies both lupus nephritis and all the rest of the lupus population that doesn't suffer from nephritis as well. CABA 201 is exciting as a product because lupus is the tip of the iceberg. And so with a fully therapeutic initial dose, one could imagine that the pursuit of additional indications could come much more quickly once we see the sort of efficacy and safety that we imagine we might see with a product that has the characteristics of this construct. Our second strategy, the CAR-T strategy, is our legacy strategy. And in that program, we have two products in the clinic. The first is DSG3 CAR-T, which is directed towards the treatment of pemphigus. And in this program, we have titrated up from 20 million up to 7.5 billion cells as monotherapy with no preconditioning. We found excellent safety, but we didn't find the level of efficacy that we would have imagined we could see. And so we initiated a combination program with cyclophosphamide plus the product. That arm of the study is now enrolling and will report out initial data by the end of the first half of this year. The MuSK myasthenia gravis program with MuSK CAR-T is currently enrolling patients, and we hope to be able to report data on that program within the next year. So in summary, three products that are IND cleared, two of them ongoing studies. The third one, CABA 201, is now IND cleared, and we're ramping up the initiation of that trial. So what are these names, these two strategies? Well, the CAR-T strategy is a strategy that uses CAR-T for autoimmunity. And specifically, what we're trying to do is reset the immune system by transiently eliminating all of the B cells in these patients. And the reason we believe this is possible is because of those publications that I referred to earlier. The CAR-T strategy with two A's is a more specific strategy. It's designed when you have a specific understanding of the epitopes that are causing the disease, and you can design into your CAR an appropriate targeting domain that will only kill about 1% of your B cell population, permanently eliminating that 1%. In both cases, the objective is to achieve a normal immune system without any evidence of clinical or biologic or serologic disease. These programs and others have now been studied across the the bolded diseases shown here. But all of these diseases could potentially be targets for a cellular therapy in autoimmune disease. It's actually quite exciting knowing that we have lupus and myositis, as well as some scleroderma data in the literature at this point. So let me go in deep on CABA 201. The academic data that I referred to earlier reveals that it's possible to reset the immune system in lupus and in myositis. The data from the papers that I referred to is summarized on this slide. On the left of this slide, you see that a 4-1BB containing CD19 CAR T product resulted in a rapid, deep, and transient CD19 B cell depletion. So you can see on the graphs, the CAR T cells expanded beautifully. The CD19 positive B cells were eliminated within a week, and the white blood cell count transiently dipped and came back up. Moving to the right side of the slide in the blue box, the clinical, serologic, and biologic responses were profound. The data was really very clear. Complete responses by 3 months in patients who were refractory to every other form of therapy. And these typically were 20-some-odd-year-old women, as is typically the case in lupus, and one man in the lupus population, and the myositis patient was also a young man. You can see the lupus, the SLEDAI scores plummet. You can see in myositis major responses within 3 months. That doesn't happen. The reporting of this data really ignited the field. And the reason it ignited the field is not only did we see this efficacy, but the safety data was profound. Because there are not that many B cells that are being killed, right? In cancer, you're killing 100% of your normals, and you're killing all of the tumor as well, the leukemia or the lymphoma cells. So orders of magnitude more than normal. In autoimmune disease, you're killing the normal 100% of your B cells. There is no tumor. And as a result, the cytokines that are released from 100% of normal B cells are apparently far, far less prominent than you see in cancer. So in this study, you saw a few patients with grade 1 CRS, 4 out of 6. That meant a fever, literally a fever. One of them in the lupus population was treated with a single dose of IL-6, and it resolved. The myositis patient was treated with a single dose of IL-6, and their fever resolved. That's the side effect profile from these 6 patients. So let's just review for a second. 6 out of 6 complete responses. Fever is the side effect. Compared to aspirin, this is pretty good. Compared to steroids, it's spectacular. And then take any of the autoimmune therapies that are used for these diseases, there's just no comparison. So as a result, the only other question you need to answer is, do the B cells come back? Because if the B cells are gone forever and the patient is now on IVIG for the rest of their life, that's not such a good solution. And in fact, because they don't have anything other than a healthy bone marrow with a selective autoimmune deficit in the post-marrow processing, that normal bone marrow is reconstituting IgM and IgD B cells, immature, naive B cells. All of the mature B cells that were pathogenic and autoreactive are gone. And what repopulates within 5 months is a normal B cell population in every single patient that has been published so far. So the data are really, really stark and incredibly exciting for patients, for physicians, and for all of the companies that are pursuing these opportunities. The icing on the cake is that these responses are durable. So it's one thing to have a complete response and have the failure of the therapy. 17 months has been publicly reported as the furthest out. All of the other patients between 5 and 17 months maintain complete response, no disease, no biologic activity, and no need for medicines. So with that data, the question is, for CABA 201, what are the prospects? Well, I mentioned earlier that CABA 201 has a similar design to the academic product that delivered those data. What that specifically means is we have an identical construct as shown here, the 4-1BB costimulatory domain, but the human binder is different. They use a murine binder that directs itself to the FMC63 epitopes. Our fully human equivalent of that binder directs itself to the identical epitopes, but it's fully human. The binding activity of the binder that we are using, our fully human binder, is similar in vitro and in vivo preclinical studies, peer-reviewed and published, as well as replicated in-house by our scientists. We know how our fully human binder is behaving relative to something that is curative and safe. If you know that and you have that data, you can now understand why we proposed and why we have pursued a trial where we might have a fully therapeutic initial dose. And the data are shown on here. Our binder also has an excellent safety profile that's really suitable for autoimmune disease. And what do I mean by that? Our partner, IASO, who we licensed the binder from, ran a study of this binder in tandem with a CD22-CD19 tandem CAR for cancer. Their clinical data showed no ICANS whatsoever in any patient at any point, probably due to the 4-1BB costimulatory domain combined with a safe binder. But that's pretty important when you're talking about perhaps the neurologic portion of the autoimmune disease portfolio. I would think it's also, and I know from talking to investigators, also really important when you think about the rheumatologic and all of the other indications. The CRS profile they reported is 5% grade 3, and everything else less than that. Some had no CRS, some had grade 1 or 2. So a really nice safety profile for the product, a lot of promise. We now need to run the study. The unmet need is clear, and I'm not going to spend time here just to save time, but it's really clear that there are hundreds of thousands of patients who would benefit from this therapy. The risk of end-stage renal disease and mortality is quite real for approximately half of those patients. Our study is, very simply put, if you have been diagnosed with any form of lupus, and you are on therapy and you are recurring or failing therapy, you are eligible to enroll. There are both the lupus nephritis and the lupus without nephritis components to our study. The objective of the study is evaluate the safety and tolerability at 28 days, but in addition, we'll be collecting the clinical activity, the biologic and the serologic findings at 3 months, and we fully expect that we should have something that replicates what we have seen in the academic literature. The timely IND filing, I didn't mention this, the IND was cleared 5.5 months after we licensed the product. And it was cleared, as I said, with what we think is a really attractive clinical program. And why is that? Well, I think it comes down to 3 things: the product, the people, and our partnership. So the product, I've gone through great detail to help you appreciate the specific way we designed the product so that it imitates as much as possible a product that is really clearly a profound clinical effect in patients with lupus nephritis. And by doing that and knowing the activity of our product and seeing the safety profile of our binder, we feel really good about the prospects. That product characteristic portfolio allowed us to pursue the clinical program that we're pursuing. The people in the company include, among other things, our chief medical officer, who led the only 2 phase 3 programs that have been FDA approved for lupus in the last 65 years. So at AstraZeneca and at Glaxo, he led both of them. So as a company, that's just exemplary, an example of the sort of talent that's in the company that has been attracted to our mission. And then I'll take to the final point here is our partnership, which is an exclusive translational research partnership with Professor Shett, which means we're not paying him to run studies for us. Rather, he is sending us his clinical samples so that our translational research scientists can give him data on his patients. At the same time, we share and we discuss the clinical findings of yet to be published data. This provides us with the opportunity to know what the future holds when it comes to the published literature. So these three things together, the product, our people and their talent, and the partnership we have, put us in a really strong position in what's going to be a fierce race to the finish line. Our manufacturing strategy is highly efficient and is shown here, has been 100% successful over the last 5 years. And as we look forward over the next months and couple of years, an awful lot of milestones and catalysts that relate to the potential for successfully curing autoimmune diseases with any one of these three products in the clinic. Thanks for your attention.